Thanks, Jen, for the introduction and welcome everyone. Thanks for joining us in this busy time of September. As Jen mentioned, today we'll be sharing some of the things that we've been up to in the Wood Lab. And won't hear actually me speak that much because I have a team of fantastic students that are actually doing this work. And so I'll give a little bit of introduction about our lab and what we do. And then you'll hear from my students as they talk about the projects that they're working so hard on. So our lab has a fairly diverse research portfolio. In beef cattle production, we span both the cow-calf sector, looking everything from calf health and calf and heifer development and cow efficiency, right through to the feedlot and the feedlot sector, where we are looking everything from efficiency and production in the feedlot right into looking at some aspects of carcass quality. But ultimately, what we're trying to do is answer important opportunities and challenges to help support a sustainable and profitable beef industry. So when we think about my research program, really has four key areas. The first one focuses on metabolic and feed efficiency. So really trying to understand why individual animals differ in their response to nutrients they consume and how they turn those nutrients into outputs like meat in the case of beef. And along with that, I also like to look at other tools and other feed management um, programs to help support some of these larger goals relating to efficiency. One of them is looking at developmental programming. And so this is the concept where either early life gestation or early life or gestational nutritional interventions can have lasting impacts on the performance and development of the offspring. And so using that concept to alter how those animals perform, again, hopefully supporting increased efficiency and sustainability for the sector. Kind of in addition, and more on the feedlot side, I also have a keen interest in rumen and gut health. And certainly with, this is a big challenge for the feedlot industry and certainly can contribute a lot to challenges in feed efficiency in those animals. So in the feedlot, we like to focus on gut health and development and see if there are ways that we can mitigate some of these challenges to support higher levels of production for feedlot cattle. As well as a classical nutritionist, so think of alternative feeds and feeding strategies, as well as new innovations in feed management to kind of help support those goals of improved efficiency, performance, and sustainability. And so throughout the rest of the seminar, you will see kind of work that we've done from my students in kind of these four key areas. Kind of in addition to some of the practical approaches that we use and the more applied scientific approaches we use, we also want to focus on trying to understand the mechanisms for some of these responses. And so whether that occurs at the whole animal level, tissue, cell, protein, right down to genetic differences. We also want to accompany a lot of the work that we do by trying to understand not only that this happens, but why this happens on a biological level. And so throughout the projects that my students will present, oftentimes you'll see some of these themes incorporated as well. All right, and again, without further ado, we have an excellent lineup of students. I'm very fortunate to have fantastic students in my lab right now, and we'll hear from them on some of the projects that they're working on. So firstly, starting off with work in the cow-calf sector, we'll first hear from Lauren Finley, who's a master's student, just starting to put the finishing touches on her thesis, looking at using gas exchange measures to better predict feed efficiency in both dry lot and pasture. And then we'll move on to looking at some projects relating to fetal programming and developmental programming. Well, you'll hear from Courtney Acton, who's my PhD student, who presents some results of a project that we did looking at protein supplementation and methionine supplementation on the impact of steer offspring in the feedlot, as well as you'll hear from a PhD student I share with Mike Steele, Corinne Hare, who is looking at the impact of starch supplementation. And she'll present some work on colostrum quality here today, but has focused a lot on kind of the impact on colostrum and insulin signaling in those cows over parturition. So without further ado, I will be quiet and turn it over to Lauren to present some of her work.
Okay, great. Thanks, Katie. Can you see my screen okay?
Looks good.
Looks good? Okay. So hello, everyone. My name is Lauren Finley. Today I'll be talking about my thesis project, relationships between feed efficiency and gas emissions in beef cows in dry lot and on pasture. So we'll start with a little bit of an introduction on beef cows and feed efficiency. So in mature cows, residual feed intake and feed conversion ratio are really challenging to estimate because they require individual feed intake monitoring, which is difficult with beef cows on pasture. And they're usually based on measurements of growth, which obviously in a mature beef cow, they remain pretty, they stay the same, obviously. So beef cows are commonly kept on pasture during the summer, where individual intake and body weight and body composition are difficult to estimate. Therefore, we need better measurements to characterize their metabolic efficiency. And so potential ways that we can do this are looking at their enteric gases. So carbon dioxide and oxygen can be used in indirect colorimetry measurements. And enteric methane is another one that's directly related to feed intake and potentially feed efficiency. So with beef cows, we see higher methane as they are on high forage diets versus concentrate. Higher intake in some cattle means more methane, and a slower rate of passage can mean more methane produced per cow as well. So a way we can measure this is with the green feed trailer. So you can see that diagram there, which basically allows short spot measurements of enteric gases from an individual animal. They go up to the machine, they put their head in, they get a little treat, and meanwhile the machine is measuring sort of their methane, carbon dioxide, and the oxygen. So we can use these measurements from the green feed trailer in measures of indirect colorimetry. So our respiratory quotient is the ratio of carbon dioxide to oxygen produced. Our heat energy is calculated from RQ. And in previous data in beef steers, it's shown to be highly correlated with feed efficiency, so a high R squared of 0.68. So there could be potential in using this for beef cows. And this may help us quantify the variation between individuals and energy use in partitioning. So the objectives of my study were to quantify relationships between traditional measurements of feed efficiency and gas emissions, rank animals by different methods of feed efficiency and compare to gas emissions and heat production and respiratory quotient, and then compare methane, carbon dioxide, and indirect colorimetry measurements from cows on pasture to dry lot measurements of feed intake and gas emissions. So we did this in two main experiments. So in experiment 1, 64 pregnant multi-paris beef cows were fed a total mixed ration. We measured their intake 24/7 for 10 weeks, and we did green feed gas measurements over 2 7-day periods. And then we measured body weight, body condition score, rump, and rib fat depth by ultrasound every 28 days. Then after they calved in the summer, we followed the same 64 cows onto pasture. So 8 groups of cow-calf pairs per group. And then we measured green feed gas measurements again over 7-day periods. And we repeated body weight, body condition, and rump and rib fat depth every 28 days. So then we calculated from the green feed measurements an estimate of heat production. And then we calculated also heat production predicted based on their intake from experiment 1. Then we ranked them by quartile groups based on a couple different traditional feed efficiency measurements. So residual feed intake adjusted for rib and rump fat depth, and dry matter intake per kilogram metabolic body weight, and contrasted these against our enteric gas emission data. So we can hop right into the results. So here is just a graph of the dry matter intake from experiment 1 compared to the heat production calculated from the green feed data. So we see somewhat of a trend between the two with the R squared of 0.2. Then we can look at our least square means of RFI quartiles. So we ranked RFI from 1 to 4, from least, so most efficient, to the greatest, so the least efficient. And we noticed that there was significance with methane. No other results showed significant relationships. And then we can look at RFI fat, so that's residual feed intake, but adjusted for rump and rib fat. And again, we saw some significance with methane in grams per day. And then we looked at dry matter intake per kilogram metabolic body weight. And again, we saw some trends with methane and carbon dioxide. And next, we correlated the dry lot and pasture gases and indirect telemetry measurements. And we saw strongly significant relationships between all of them except respiratory quotient. So these are between experiment 1 and 2. So in conclusion, our traditional methods to quantify feed efficiency are not ideal for mature beef cows. Methane could be a valuable indicator of feed efficiency, but further research is needed. Gas emissions can be measured consistently between dry lot and pasture. I'd like to give a thank you to our sponsors and the staff at the Lower Beef Research Center and the volunteer assistance from members of the Wood Lab.
All right, thanks so much, Lauren. And well, I guess we'll hold all the questions to the end. So now we'll switch gears a little bit and talk some more about some of our developmental programming projects. So Courtney Acton will be up next, sharing some results looking at protein and methionine supplementation on steer offspring performance. So Courtney, if you can share your slides.
Okay, I'm hoping you can see it.
Looks good.
Awesome. Okay, so like Katie said, I'm going to be talking about my master's research, where we looked at how cow nutrition during pregnancy can set a calf up for success. So the Barker hypothesis explains that undernutrition during key developmental periods in pregnancy can reduce fetal growth and create a scenario of metabolic mismatch. This is when there's a nutritional difference between the in utero and postnatal environments, and that can alter the development of muscle and fat for the calves. Severe malnutrition has negative implications for the cow and calf, which isn't ideal, and it can result in reduced income and increased cost for the producer. Previous studies have used a global restrictive diet, which reduces the amount of energy and protein that's provided to the cow. So if a mild protein restriction while maintaining energy can beneficially influence steer calf development and carcass quality, it could be a helpful management tool to increase the revenue for Canadian beef producers. During late gestation, cows have increased nutrient requirements to support their growing calf. Previous studies have looked at increased crude protein content in cow diets, and they've reported increased weaning and slaughter weights of male beef calves, as well as improved carcass characteristics, including marbling and quality grade. But there hasn't been a whole lot of research conducted to investigate the effects of maternal metabolizable protein content on beef steer offspring throughout life. In beef cattle, methionine is commonly considered a limiting amino acid, and it's involved in many metabolic pathways in the body. Methionine supplementation has been shown to alter gene expression in offspring. Maternal supplementation in the beef and dairy industries have resulted in different outcomes. So dairy calf body weight and growth was improved between birth and weaning weight at 8 weeks of age. But beef calves show improved average daily gain at weaning at 6 months of age. Currently, there's a lack of research investigating methionine supplementation during gestation on feedlot steer growth and development past the weaning period. So timing of nutrient manipulation is key. The majority of fetal development occurs in late gestation, which is also where muscle fiber maturation and fat development also occurs. Nutrition during pregnancy can influence the offspring's development of muscle and fat. So dietary manipulation, specifically during late gestation, can have a lasting impact on offspring development and meat quality. We think that methionine can alter calf growth and production outcomes. And we also wanted to evaluate how a mild protein restriction impacts calves. So the main objectives of this experiment were to examine the effects of maternal nutrition in late gestation on offspring growth and feed efficiency in the feedlot, blood metabolite concentrations, carcass and meat quality characteristics, and the expression of genes related to growth, muscle development, and fat accumulation in the offspring. In a research-intensive study, 138 Angus cross cows at the Elora Beef Research Center were included in a nutrition trial to assess how metabolizable protein levels and methionine supplementation impacts gestation. In a 3x2 factorial design, cows were randomly assigned to one of 3 protein treatments, feeding to meet 90, 100, or 110% of metabolizable protein requirements for cows during late gestation. Half of each group was top dressed with pellets containing 9 grams per day of rumen-protected methionine. Palm fat was also added to the diets to ensure that the energy content was consistent. There's currently no research evaluating how maternal methionine impacts calves in an industry-applied setting. So a second trial of 67 Angus cross cows at the New Liskeard Agricultural Research Station were randomly assigned to control our methionine treatments. In addition to unlimited access to water and hay, cows were fed once daily in a bunk. They received either 0.75 kilograms per head per day of supplemental pellets supplying 12 grams per day of rumen-protected methionine per cow, or a similar pellet without additional methionine. Cow diets at both locations were fed for approximately 8 weeks prior to calving. For the present study, 55 steers from Elora and 34 steers from New Liskeard were transported to the Elora feedlot facility. All steers were managed as a group, and they were fed corn-based diets formulated to meet the requirements for growing and finishing beef steers. Collected samples included feed and individual feed intake data using the Incentec feeding systems, body weight, blood to assess metabolic profiles of individual animals. Carcasses were assessed by a certified grader for fat and lean yield, and rib dissections were used to evaluate body composition. Steaks were also used to assess color, pH, and tenderness. Neck muscle was collected at slaughter for RNA isolation and analyzed for genes related to growth and development of fat and muscle using real-time PCR. It's important to note that Elora and New Liskeard data were analyzed separately. So steers from cows fed 90 and 100% MP had higher non-esterified fatty acid levels in their blood, which indicates altered fat metabolism, potentially from visceral depots. Additionally, steers from cows fed at 90% MP also had higher grade fat. So in conjunction with the elevated NEFA levels, these findings support the Barger hypothesis, specifically the metabolic mismatch, which states that undernutrition during gestation, followed by diets that meet requirements postnatally, influences fat development. Steer growth was different between management systems. Steers from the industry-applied study had consistently heavier body weight. This could be due to the New Liskeard cows being unrestricted in the amount of feed that they could eat. Inclusion of methionine in cattle diets has been shown to increase feed intake. So if cows eat more during pregnancy, this would allow for increased nutrients to be partitioned towards their growing calf. In the industry-applied study, methionine supplementation reduced the amount of drip loss as a result of cooking the steaks. As to be expected, the longer that the steaks were aged, the more tender the meat was, and the less moisture was lost during the cooking process. However, maternal diet did not impact the majority of steer offspring carcass and meat quality measurements. Although it's not significant due to our low animal numbers, the majority of our steer carcasses were graded as AAA. Steers from cows fed over protein requirements had increased expression of myogenin, which is important for the differentiation of muscle fibers. Previous studies showed differences in myogenin response from maternal nutrition. Fetal tissue showed increased myogenin expression when cows were energy restricted, while early weaned heifer calves showed no differences when cows were oversupplied metabolizable protein in late gestation. This shows that myogenin expression could differ based on the age and sex of calves. Steers from cows without methionine supplementation had increased pyruvate kinase. Pyruvate kinase is the final step in glycolysis, and it's recently been shown to increase tissue growth and muscle development. Although research in methionine's effect on PKM is minimal, its expression has been shown to increase in calves as they age, and could be due to an increase in glycolytic capabilities. Interestingly, PKM expression in muscle from steers in the industry-applied study was opposite from the research-intensive study, with offspring from methionine-supplemented cows in New Liskeard having a higher expression of pyruvate kinase. This suggests that the offspring response could differ based on maternal management practices. Perhaps the increased expression of PKM may be due to the cows eating more feed during the last 8 weeks of gestation. Presence of methionine has been shown to enhance pyruvate transport and use towards glycolysis, which could lead to increased tissue growth and muscle development, resulting in increased PKM expression. Myogenic differentiation is another regulatory factor involved in muscle development. Maternal supplementation of methionine increased the expression of MyoD in steer offspring muscle. There's not a whole lot of research that's available to assess methionine effect on MyoD, but its expression was increased in calves from energy-restricted dams. Overall, these studies have demonstrated that maternal nutrition in late gestation has an important impact on steer offspring throughout life. Body weight was increased in the industry-applied study, which may be due to the differences in cow feed intake. Feeding cows below MP requirements in gestation increased serum NEFA levels and grade fat, which supports the metabolic mismatch of the Barker hypothesis and suggests that fat was altered. Methionine supplementation in the applied study reduced the amount of moisture lost through the cooking process, and the aging of steaks improved meat tenderness. Maternal nutrition increased the expression of genes that are key in muscle development, and cow management may contribute to the opposing differences seen in the expression of pyruvate kinase between the two studies. It's clear that calf success starts with cow nutrition. Growth and fat development may be able to be improved in calves from cows that experience nutrient restrictions during late gestation, if the calf is provided a diet that meets requirements postnatally. The effects of maternal nutrition were different between the studies, illustrating that offspring development can vary depending on management practices. And producers may be able to manipulate nutrition in late gestation to support muscle development in the calf, which could increase producer profit, improve meat characteristics, and lead to quality AAA Canadian steaks. Thank you to everyone who made my master's research possible.
Great, thanks so much, Courtney. And again, a little bit of taste of some of the developmental programming work we've been doing relating to protein supplementation. And now I'll turn it over to Corinne, who is, again, a PhD student that I share with Mike Steele, who is going to share some of her work looking at the energy supplement side.
I did. So yeah.
Oh, Jean, I think your mic is on. Go ahead there, Corinne, if you can share your slides.
Absolutely. Can everybody see everything okay?
Looks good.
Okay, great. Thank you. Thanks for the introduction. So the title of this research is Late Gestation Metabolizable Energy Intake is Increasing Colostrum Yield and Altering Colostrum Composition in Beef Cattle. And so to preface a little bit, we started with this idea of late gestation glucose balance that comes from looking at the cow's metabolizable energy requirements during late gestation. They'll generally be about like 75% greater than a cow's similar body weight that's non-gestating. And if we really zero in on the substrates that are being used, she has a large glucose draw because about 50 to 60% of the oxidative substrates for the fetus are coming from glucose and lactose, or pardon me, lactate. And so we were questioning how that would look relative for supply relative to demand, especially with the data that comes out of this overfed dairy cow model, in which it was predicted that in the week preceding calving, there's initially a leveling off of supply relative to demand for glucose. And then about 3 days prior to parturition, there was actually a glucose deficit. And so this could be significant for colostrum production because glucose is going to be that precursor for lactose synthesis. And then essentially in theory, if we can increase lactose production, we can increase colostrum yield. And so this was demonstrated using twin and singleton bearing ewes in 2004 by Banchero et al., in which they oversupplemented the ewes prior to parturition using a ruminal bypass starch source, and they managed to have more than a doubling of the colostrum yield. But they also altered the colostrum composition. They decreased the fat content and the protein content while increasing the lactose content, which would be indicative of that osmotic drive. But similarly, in dairy cattle, when they were overfed energy relative to their requirements during late gestation, there was a decrease in IgG content. And so collectively, this data is indicating that metabolizable energy content of the dry ration is going to be having some influence over the process of the colostrogenesis, and that the amount of glucose that is supplied to the cow could be having a role in the yield of colostrum. But there's not much data on this relative to beef cattle, and if we contrast rations between the overfed dairy cow model compared to a late gestation beef cow, we'll see that they're more forage-based rations that are less energy dense and have fewer substrates that contribute to the cow's glucose pool. And so from this, we were asking the questions, do beef cows experience an antepartum glucose deficit? And can we modulate beef cow colostrum production through her metabolizable energy intake during late gestation? So this brings us to the hypothesis that increase in late gestation metabolizable energy supply would correct probable deficits in antepartum glucose balance to increase colostrum yield and alter colostrum composition. And we had the objective to evaluate how increasing late gestation metabolizable energy supply would impact colostrum yield and composition. So we ran this study. We started in January of this year at the Ontario Beef Research Centre using 156 Simmental Angus crossbred beef cattle. We had 47 heifers and 109 cows. And then we assigned them to 3 treatments about 53 days before they were about to calve. And these treatments were designed to supply about 80%, 100%, and 120% of their metabolizable energy requirements. And as we stepped up the energy density of the ration, we were increasing the concentrate allocation and substituting in a starch source that would be bypassing the rumen for digestion in the small intestine. At calving, we were looking at colostrum composition, colostrum yields. So from pretty much all of the cows, we were able to collect a colostrum sample. And then in a subset of the cows, we were milking colostrum right after calving. And we did this by attaching a portable milking machine right at calving and injecting 2 cc of oxytocin. And then we would measure that full yield once the pump stopped extracting colostrum from the udder, subsample that, and then feed what we had collected back to the calf. And this was all done within about an hour of the cow's calving. We were predominantly interested in the composition and the IgG concentration of the colostrum samples. And so we were looking at the fat, the protein, and the lactose. And then IgG was measured by Saskatoon Colostrum Company. So we achieved our goals. We managed to increase colostrum yield when we stepped up the energy density of the ration. So we see that treatment affected the colostrum yield with high metabolizable energy cows producing more colostrum than the low metabolizable energy cows. And then we have this effect of parity. That's not unsurprising because we do know that multiparous cows in general produce more colostrum compared to the primiparous cows. For composition, We see that crude protein decreases as the energy density increases, and then we see a similar mimicking trend for colostrum urea nitrogen, as well as colostrum BHBA concentration, where they are decreasing as the energy content increases. In terms of lactose, we saw, as we were hoping to see, and it mimics what was seen with Banchero et al., where we have an increase in lactose concentration of the colostrum as we increase the amount of starch that's provided in the diet. And for the crude fat concentration, these results are a little bit less in line with what was seen before. So we do still have a decrease in the colostrum fat concentration between the low and the control. But contrary to what we were expecting to see, the high metabolizable energy cattle actually produced colostrum that had the same fat content as the low and the control metabolizable energy treatments. And so we're initially speculating that this might be due to stimulating insulin resistance by overfeeding energy during the dry period. For colostrum IgG concentration, our results are similar to what was seen in dairy cattle out of Cornell in 2016, where we have a decrease in colostrum IgG concentration as we increase the energy density of the ration. But because those cows were producing more total colostrum, we have a greater IgG yield with the high metabolizable energy treatment compared to the low metabolizable energy treatment. And though there was no difference due to parity for the total concentration of IgG, there was an effect of parity in regards to yield where the multiparous cows produced more total IgG compared to the primiparous cows. And I will note that in all scenarios, regardless of the treatment or the parity, those calves in the first meal are consuming above the recommended thresholds for IgG mass intake during the first hour of life. And if we consider this relative to some data that was just published this year out of Cornell, that initial meal is probably representing about 25% of the mass of IgG that calf is going to be consuming within that first day. Which we think is reflected in the calf serum IgG concentrations, as we're seeing that by 12 hours, they're already up above 45 grams per liter, which is already above the threshold that's recommended for beef calves at 26 grams of IgG per liter. And then they're peaking roughly at about a day, and then they're on decreasing with no differences due to the treatment, probably because the gut has been saturated by the amount of IgG that they're intaking. So with this data, what we can conclude is that in regards to colostrogenesis, it's pretty clear that late gestation metabolizable energy supply is impacting beef cow colostrogenesis. And regarding calf immunity, we might say that since beef cows are producing such substantial amounts of IgG, manipulating the colostrum volume and IgG content will have minimal impacts on beef calf serum IgG. All right, and so with that, I will pass it back to you, Katie.
All right, thanks, Corinne. That's great. I'm just going to share my slides again here. And so switching gears a little bit, moving away from cow-calf research and shifting into the research pertaining to the feedlot sector, I'd like to introduce my next two students. Amber Zupan is a master's student, and we just finished her trial looking at the impact of undigestible nutrient detergent fiber and starch fermentability on steer performance and gut health. And then we'll hear from Melissa Williams, who's another of my PhD students, who has been doing some modeling work looking at the impact of UNDF on steer performance. And so, Amber, if I will stop sharing here and turn it over to you to hear an update on the progress of your work. So if Amber, you can share your slides. Oh, Amber, are you there? I can't hear you or see you, Amber. Maybe we'll switch if Amber's not there, and Melissa, if you want to start, maybe?
Yep, I can share my screen and get started if you want.
Sure, maybe we'll do that. So we'll start with Melissa talking about her meta-analysis.
So can everyone see my screen here?
Yes.
Okay, perfect. Okay, so as Katie said, my name is Melissa Williams, and I'm a PhD student under Katie. I've been working on a couple different side projects lately. I've actually been working on 2 different meta-analyses, so I'm going to give you an overview of the one that I have finished and a quick introduction to the second one that I'm starting right now. In essence, for any of you who do not know what a meta-analysis is, it allows us to answer a specific question with an increased power by extracting data from the literature in order to make conclusions on a set of similar experiments. So my first project is looking at UNDF as a predictor of feedlot cattle performance and carcass characteristics, in which traditionally finishing feedlot cattle are fed high-grain diets to promote growth. However, this does increase the risk for ruminal acidosis, which is associated with loss of producer profit due to health issues and reduced animal performance. To aid in preventing acidosis, some forage is included in the diet to increase the amount of effective fiber to increase rumination and saliva production. This is typically formulated by feedlot nutritionists on a forage inclusion basis. It is important to note that not all forages are considered equal, as they are made up of different amounts of digestible and indigestible nutrients, which can differ based on crop harvesting, processing, and chemical treatment. Neutral detergent fiber is a portion of the forage that is included in diets. However, just like forages, not all NDF is considered equal either. Undigestible NDF, which is a portion of the NDF, is never digested or absorbed for use by the animal. This non-nutritional component of the diet can be considered a constant measurement in comparison to forage inclusion or NDF percentage, and has the potential to be used as a predictor in animal performance and carcass characteristics. It is important to note that UNDF is extremely underreported in cattle nutrition experiments, and to date there are only 2 published experiments that examine UNDF concentration in the diets of feedlot beef cattle. An experiment examining lactating dairy cow feeding behavior, fiber digestibility, and lactation performance found that increasing dietary UNDF from 9.5 to 11% reduced milk protein and lactulose, reduced UNDF intake and time under pH thresholds, and NDF digestibility. No differences were found between the 2 UNDF levels on dry matter intake, rumination, or average pH. An experiment focused on feeding behavior, performance, and ruminal characteristics in beef cattle found that UNDF, increasing from 4.6 to 5.6%, was found to increase UNDF, It was found that increasing UNDF, where there was a decrease in eating time, sorting against long particles, and proportion of acetate in the rumen. Increasing UNDF in the diet also decreased total volatile fatty acid concentration, duration of pH under thresholds, as well as reduced dry matter, organic matter, and crude protein digestibility. The second and only other experiment done in beef cattle found that decreasing UNDF from 7.8 to 6.7% had no impact on dry matter intake, feed conversion, and increased dressing percentage, and made changes to the carcass yield grade proportions. Here, it's important to note that in these experiments, UNDF only differs between 0.5 and 1.5% on a dry matter basis. A meta-analysis would allow for the examination of UNDF impacts, not only within study, but across studies, which would increase the range of UNDF values we could examine. So that brings us to this project, where the objective is to conduct a meta-analysis to look at the relationship between dietary UNDF in finishing feedlot cattle diets, on animal performance and carcass characteristics. Our hypotheses were that increasing the inclusion of UNDF would reduce the average daily gain while increasing dry matter intake to meet nutrient requirements, resulting in poorer feed efficiency and reduced carcass characteristics. Additionally, it was hypothesized that dietary UNDF would be a stronger predictor of animal performance and carcass traits in comparison to NDF. So for this project, we collected data from studies that examined forage type or forage inclusion as treatments in finishing feedlot beef cattle diets. We collected a multitude of different variables that could potentially be used in models down the road. And then we also used the Cumberland Valley Analytical Services database in order to estimate the UNDF percentage of each of the dietary treatments. From here, we began working on model development. So we started looking at our outcome variables of interest, which for performance included average daily gain, dry matter intake and gain to feed ratio, and carcass characteristics of final body weight, hot carcass weight, and dressing percentage. Our potential drivers for this project, namely, were UNDF and UNDF intake, which had to be a part of our models in order to move forward in the model evaluation phase. This is due to our objective that we want to understand UNDF better in these outcomes. Additionally, we had other variables that could be added to the models if significantly important to these models, including things like days on feed, whether or not implants or other feed additives were used, and chemical composition of the diets. Once models were evaluated and determined to be statistically significant, the models were then evaluated in order to determine which model was best for each outcome variable. The evaluation statistics included information on model bias, deviation, disturbance, precision, accuracy, and shifts. So this brought us to our final models, one for each of our outcome variables of interest that you can see here on the slide. Here you can see when looking at UNDF variables in each of these models, UNDF had a negative impact on all but dry matter intake. This should make sense because as we increase the amount of undigestible nutrients within the diet, this would force an animal to consume more in order to meet their nutritional requirements and reduce their overall feed efficiency. This would result in slower growth and potentially reduced outcomes at slaughter. So overall for this project, it was concluded that the models that were discovered are pretty easy in terms of being used for feedlot producers and potentially nutritionists as well. And this is because the variables that are needed in these models, other than UNDF%, which can be tested for, are fairly simple. And a lot of producers do know things like dry matter intake for their animals. And additionally, implant and tylosin, which can be added to these models to improve their prediction, is only a yes or no answer. In order to improve it. So we don't need to know when you're implanting, what type of implant you have, or how many times you're implanting. We just need a yes or no answer in order to improve that outcome variable prediction. Additionally, it was found that UNDF was a better predictor of these outcomes in comparison to NDF. And overall, we recommend that testing and measurement of UNDF in the diet should be increasingly adopted in both research and in the field in order to better understand this component of the diet and how it impacts beef production. In terms of next steps from here, we'd like to examine the effects of UNDF on the rumen. So we were able to look at a whole animal model in terms of animal performance and carcass characteristics, but the rumen does play a big role in this as well, so it'd be a good thing to take a look at. Additionally, again, we can look at how UNDF impacts ruminal kinetics and how that in turn impacts our outcomes that we saw here today. Additionally, some future research should be done in animals that are potentially challenged. So all the data that was collected in this project were on healthy animals that were not challenged, for example, under a ruminal acidosis state, in which our UNDF may or may not have a different impact on that animal depending on what kind of stress or challenge they're under. So now just a quick overview of my second project in which I'm looking to improve the prediction of feedlot cattle ruminal pH. So there are several models currently out and being used to predict ruminal pH in cattle. However, only a few are developed using actual beef cattle data, not dairy cattle data, in which it was determined by some researchers back in 2015 that the beef models that were best in terms of reliability for ruminal pH prediction were the ones that were developed on beef cattle data sets. So the current models use physically effective neutral detergent fiber, or PENBF, or total volatile fatty acid concentration, TVFA, as driving variables in their models. So these models are great. However, we do know that there is some room for improvement, and this is because there is a multitude of variables that can impact ruminal pH, and our current models are only looking at 2 of them. So this provides a bit of room in terms of trying to improve our current models to better predict the outcomes that we're interested in, which is ruminal pH. So the objectives of this project are to examine the current recommended beef cattle ruminal pH models using the new data set that will be developed during this meta-analysis, and then determine new driving variables for the prediction of ruminal pH that could potentially improve the current models. So far, we have just finished the data collection phase, in which we collected data from studies that examine finishing feedlot cattle diets, where the forage type or inclusion were the treatment, and where they measured ruminal pH, volatile fatty acid concentrations, and PE NDF where possible. Thank you very much.
All right, thanks so much, Melissa. I did get a text from Amber saying that her computer froze, but hopefully she was able to rejoin. Amber, are you there?
Yes, I am back. Hopefully you can hear me.
Excellent.
Sorry about the technical
you.
difficulties, guys. Thanks
All
for
right.
jumping ahead, Melissa.
All right, Amber, if you can go ahead and share your slides. Perfect.
Right.
You can see them.
You can see them okay?
Yeah.
Perfect. Okay. So hello, everyone. My name is Amber Zupan, and the research that I'm presenting today will be looking at determining the impact of undigestible NDF and starch fermentability in feedlot steers on performance and gut health. So the project that I've been working on is actually a cluster project with the goal of determining the minimum fiber requirement for feedlot cattle and improving the empirical prediction of rumen pH. We're actually fortunate enough to be a step in this project with partners at the University of Saskatchewan, the University of Lethbridge in Alberta, and the University of Guelph. This cluster project does contain 4 phases with a total of 6 studies between the phases. And the trial that I will be discussing today is a part of phase 3, which is using undegradable NDF and ruminally fermentable carbohydrates to optimize diet formulation for feedlot cattle. So the typical North American diet contains 8 to 10% forage inclusion, and forage helps to promote chewing, which increases salivation, ultimately aiding in the regulation of rumen pH. Additionally, forage promotes rate of passage from the rumen, and inclusion rate can be used to predict rumen pH. However, it doesn't really consider the effectiveness or digestibility of the fiber. Since not all forages are created the same, it's important to investigate the effects of the composition of the forage. So NDF stands for neutral detergent fiber and includes the cellulose, hemicellulose, and lignin portion of forages. One type of NDF that falls in this category is PE NDF, which is physically effective NDF. PE NDF is the physical form of NDF in the diet, which is referring to the quantity of forage fed. Another type, which we'll be focusing on in this project, is UNDF, or undigestible NDF, which is the concentration of NDF in a forage, referring to the chemical quantity within the fiber. UNDF is often of great importance because it's been recently incorporated into nutritional models and is believed to have a direct relationship on ruminal motility, rumination, and thus ruminal function. NDF functions as a part of the rumen mat, which also contributes to ruminal motility. When there's increased UNDF, there will be increased rumen mixing, allowing for more absorption potential and ultimately resulting in a more easily regulated rumen pH. In addition, when the amount of grain is increased by replacing forages, cattle will become at risk for metabolic disorders. Acidosis is a common problem that will develop when fed rapidly fermentable carbohydrates due to the accumulation of total organic acids and lactate in the rumen. This will eventually result in a decrease in rumen pH, leaving the gut epithelium compromised. Once this gut wall is compromised, this allows for bacteria to escape from the rumen and enter the bloodstream. These bacteria will then travel through the blood to the liver and may result in liver abscesses. Liver abscesses are currently being looked at in reference to the quantity of acute phase proteins in the blood, which can be a marker of inflammation. So the objective of this study is to investigate the effects of grain fermentability and UNDF content in the diet on animal efficiency and performance, feed intake and feeding behavior, and rumen pH. We hypothesize that as UNDF increases, ruminally fermentable carbohydrate increases, it will improve rumen pH, feed intake, performance, and efficiency. In addition, we hypothesize that as ruminally fermentable carbohydrate increases, the requirement for UNDF will also increase. So in this 96 Angus crossbred yearling steers at a weight of approximately 500 to 550 kilograms, all located at the Ontario Beef Research Station, were started on trial. The steers were blocked by body weight into 2 groups and were started 3 weeks apart in order to ensure not all steers were finished at once. Steers were randomly assigned to 1 to 12 pens with 8 animals per pen. The steers were then randomly assigned to their treatment diet, where 2 of the 4 treatment rations are fed per pen, yielding 6 different diet pen combinations per block. Steers were trained to use the assigned bunks and adapted over a 4-week step-up period to the high-grain treatment rations by increasing the ration by 20% per week and decreasing the forage in the grower ration. This study was designed as a 2x2 factorial design with grain fermentability and UNDF as treatment factors. The treatment diets included either a highly ruminally fermentable grain-based diet or a low fermentable diet with either a forage high in UNDF or a forage low in UNDF. This yielded 4 treatment diets, so corn silage and rolled corn, which was our low-low diet, corn silage and high moisture corn, which was our low-high diet, haylage and rolled corn, which is our high-low diet, and haylage and high moisture corn, which is our high-high diet. The diets were formulated to be both isocaloric and isonitrogenous while keeping the forage inclusion at approximately 5%. All treatment diets included a similar pelleted supplement containing soybean meal, urea, limestone, and a commercially available trace mineral mix with 33 parts per million of monensin. The diet composition and nutrient values can be found in these tables shown. On day 1, steers were weighed on consecutive days to normalize for gut fill and every 28 days thereafter. In addition, half of the steers, sorry guys. I'm so sorry, my throat's so dry. In addition, half of the steers also received an indwelling pH probe to measure rumen pH and temperature 4 weeks before slaughter. Steers all started with an approximate body weight of 550 kilograms and were fed for approximately 100 days until reaching the target market weight of approximately 750 kilograms. Steers were chosen based on the highest body weight to go to slaughter at the University of Guelph Meat Lab, and 8 to 12 animals per week were slaughtered to facilitate sample collection. So throughout the trial, various data has been collected looking at animal performance, feed intake and feeding behavior, and gut health. Performance traits evaluated will include average daily gain, feed efficiency, and hot carcass weight. Feed intake and feed behavior measurements were tracked by the Incentec feeders, which are able to show individual meal size at each time the steer enters the bunk, as well as time spent in the bunk by reading the RFID tag in the steer's ear. Additionally, we collected orts to assess feed refusals, which we are working on processing through the Rotap sieve shaker and Penn State particle separator to assess particle size differences. At 28 days on trial and 28 days before slaughter, as well as the day before slaughter, a fecal sample and blood sample was taken to assess fecal starch and acute phase proteins in serum. In addition, 28 days before slaughter, an indwelling pH probe was inserted into half the steers to continuously measure rumen pH for the last 4 weeks before slaughter. At slaughter, liver abscess scores were assessed using the Elanco scoring system. In addition, we assigned a rumen health and intestinal health score on a scale of 0 to 3 and collected rumen and cecal fluid for pH and VFA analysis. Finally, the carcasses were graded by a certified Canadian Beef Agency grader. This research is important as current nutritional models are also starting to incorporate UNDF, which is believed to have a direct relationship on ruminal motility, rumination, and hence ruminal function. While there's a large importance to incorporate UNDF into these models, there's currently little research looking at the effects of UNDF and the minimum fiber requirements on various parameters in beef cattle. I have to present today as our trial just ended in late August, but I look forward to sharing my results with you in 2022. Thanks for being here today.
All right, thanks, Amber, and go get yourself a drink of water. So yeah, as Amber mentioned, we just finished this experiment, so you'll have to stay tuned for a future seminar to find the results of Amber's trial. All right, so to finish up here, I'll just share my last slide. So as you can see, these 4 kind of key principles are used quite heavily in my research program, but also there's a lot of interaction between these concepts. So it showed you a little bit of a taste of what the work that is going on in my lab right now, but we're doing quite a lot of work in rumen health and gut health. Melissa will be doing some work looking at direct fit microbials in feedlot health, as well as Courtney will be doing some work looking closer at metabolic and feed efficiency on the cow side. Also, just as a little bit of an advertisement, my master's student, Vanessa Rotondo, defends tomorrow, and she did a kind of a unique project looking at the relationship between linear body measures in calves and the correlation with body weight, as well as the ability to predict weaning weight from those early life measurements. So she did some unique things, including looking at machine learning approaches to analyze this data. So if you're around tomorrow, feel free to join us for Vanessa's presentation in defense. Certainly, as you can see, we had a pretty active group, and I have some fantastic lab students. So thank you again to all my wonderful lab students, past and present. Certainly make me look really good. As well, we couldn't do any of this work without the support from the staff at the research stations, both the Laura New Liskard, as well as the Meat Lab staff, as well as technical support from Tim and Julie has helped us out on some of these projects. We have a lot of collaborators on this work, and I won't name them all, but certainly I appreciate all your thoughts and insights into these projects. And of course, we can't do any of this work without the support of the industry and the funders of this research. So thank you to everyone who has invested in our program, and hopefully seeing some changes towards, again, that sustainable and profitable beef industry. And so with that, Jen, I think finished almost exactly on time, which is a bit of a small miracle. And so happy to take any questions, either for myself or any of my students from the audience. Great. I'll just exit out here so I can turn my video back on.
Thanks so much, Katie, and all of the grad students. Really nice job. We'll open the floor for questions. You can just unmute yourself and speak up, or you can type your question into the chat. So any questions? Christine, you have your hand up, I think.
Yeah, I have. I think it's great. What a fantastic palette of great research. So kudos to you, Katie, and to all of your students as well. I have some questions about the feed efficiency work that's being done. We're doing not similar, but sort of similar work in dairy, and I've been wondering for a long time if it's possible to include activity of the animals as a source of energy loss or expenditure. We've got heat, we've got feces, methane was mentioned, growth was mentioned, milk is mentioned for us in dairy. But what about activity? Do you guys see that as a real part of the energy that's being used?
Yeah, so great question, Christine. And certainly it's something that we're not looking on specifically, but others have researched kind of the activity level. And so there's work out of Australia that kind of estimate how much activity contributes to individual animal differences. And it's actually relatively small, like maybe 8 to 10% between individual animals. Mm-hmm. That being said, certainly, particularly with cows on pasture, there is a significant maintenance energy increase relative to the activity level on pasture. And so we know that things like in years where forage availability is low and animals have to travel more to graze, the maintenance cost of those animals increases. And so as part of the NRC model, there is adaptions for energy expenditure relative to grazing. But that would be like on the the whole animal level. But with the individual animal differences, it's supposedly, according to the research, relatively small difference based on activity level.
Okay, cool. Thanks for that. I have another question. Can I do it? Can I? Jen is nodding.
You have to ask
Go
the
for it.
taskmaster here, Jen.
Go for it,
She nodded at me, so it's good. So I'm really interested in the genetic differences between these animals, and you've got some really nice experimental designs where everything is sorted out already, and you've got your numbers all nice. I'm thinking about Corin's experiments and Lauren's experiments, and actually the experiments of the whole team. Have you looked at all at like using a daughter design or a granddaughter design overlaid on top of your experimental design to see if there's differences between the different sire families that were used?
Yeah, so for us, the genetic differences end up falling into that error category. And so they end up being part of the random error. So we tried to minimize that a little bit by kind of minimizing the number of sires that we have. So at least in both Corinne's and Courtney's study, there was only about 5 or 6 sires used across the herd. But presumably we could evaluate that, and maybe we should chat later. And I know that Angela has DNA sequenced on some of them. I'm not sure if everyone has DNA sequenced, but there definitely are other genetic components that can play in here. With relatively smaller numbers on these specific designs, it might be tough to get at that response.
Well, I mean, if you've got a relatively small number of bulls, and if you've got a really nice spread of those bulls across your treatments, even without genetic information, just with a straight up granddaughter design or daughter design, I think you could start to see if there's differences between the the different sires, and then it would be like a really nice additional it's a bit of a sidetrack, but it's a nice additional ice cream cone, so to say, from all of the great research that you're doing anyway. So if you're interested, we can chat, and I think that would be cool.
Sure, sounds good, Christine.
Thank you. Great work.
Thanks.
Awesome. Any other questions for Katie or her group?
Hi, I have a question. This is Angie. This
Hi,
is
Angie.
a question. Hi. For Corinne or about Corinne's stuff. It's more of a methodology question. Did you say that you measured colostrum quality using the Saskatoon Colostrum Company?
Yeah,
sure,
so-
I'll turn that over to Corinne. Go ahead, Corinne.
Okay.
Yeah, so IgG content was measured by Saskatoon Colostrum Company, and that would be done by radial immunodiffusion.
Okay. All right. I noticed that the paper you referenced from Cornell published this year, they made quite a case for not
for
using
looking at
bricks.
the abundance. Yeah.
Yeah. Okay. Thank you.
For sure. You're welcome.
Yeah, we've got some other data that it didn't present here that kind of compared IgG with radiodiffusion, BRICS values, and then we also used an activity assay using a rabies vaccine challenge model. And so they were actually fairly well correlated, although the BRICS seems to account for other things, not just IgG. And so, yeah, BRICS is a good, inexpensive indicator, but it is not the gold, should not be the gold standard, because it could mean some other things as well.
Okay, thank you. And is that pretty well the opinion in the beef industry, or what are the thoughts right now?
Well, to get producers to think about colostrum at all is a kind of a newer concept in the beef industry. Certainly they know that colostrum is important, but we're learning more things about the importance of colostrum, even additional things not related to IgG. So there's a laundry list of bioactives that are in colostrum, and we've done a little bit of work here too. And Corinne and Mike are also working deep in this area, looking at, can we modulate some of those bioactives with nutrition prepartum, and what impacts the levels of those bioactives have within the colostrum on future calf performance. So there's a lot of hormones that are different in colostrum, like insulin, which Corinne has done a lot of work on, even IGF, prolactin, lots of others that could have some pretty significant implications on calf development. So yeah, working kind of closely with Mike and his group, we're looking at this on the beef side as well.
Okay, awesome.
Great.
Thank you for the information and thanks for a wonderful presentation.
Yeah, great. Thanks, Angie.
All right. Well, I'm cognizant of the time, so maybe if you've still got burning questions, reach out to Katie and her students directly. I'd like to thank Katie and all of her grad students for their nice presentations today. And we will resume our CNM seminar series in October, where I believe we have Trevor DeVries and his lab presenting. So you will see an email communication and invite on that in the next short bit. So with that, I'll say thank you guys all for joining, and we'll see you again next month.
Thanks, Jen. Thanks,
Okay.
everyone, for joining.